Hello friends, welcome back to in my YouTube channel that is BiologyCenter. Today I am going to teach you classification and nomenclature of enzyme. Okay, so before starting this topic, first of all you have to know the term that is what do you mean by classification and the nomenclature of enzyme. Okay, classification means that is grouping of, please do remember, grouping of anything, please do remember, grouping of anything into convenient category. Okay, please do remember, grouping of enzyme, I mean anything that is enzyme, anything means that is enzyme, I'm talking about enzyme. Okay, grouping of anything that is enzyme into convenient category. Okay, that is called classification of enzyme. Okay, but when you're talking about nomenclature of enzyme, okay, then it simply means naming of enzyme. Okay, naming of enzyme that is accordingly that is 4 digits number. Please do remember, naming of enzyme accordingly 4 digits number. For example, when you're talking about and this name is given by that is IUBMB. Please do remember, in case of chemistry, when you're talking about the name is given by IUPAC, but when talking about enzyme, the name is given by IUBMB. Okay, I stands for, if you remember, International Union of Biochemistry. International Union of Biochemistry, Molecular Biology. Okay, now International Union of Biochemistry and Molecular Biology. Okay, these 4 digits number. Okay, the enzymes name are given on the basis of 4 digits number. For example, let's see. For example, talking about an enzyme, then you have to write down EC. Okay, that is enzymatic. It stands for enzymatic committee. Okay, now for example, that is 4 digits, but 1, 2, 3, 4. That all 4 digits are separated by dots. Okay, that is first digit represent. Please do remember, first digit represent class. Okay, and the second digit that represent subclass. Okay, and the third digit represent subclass. Okay, it may be divided into subclass, and the last digit represent order, that is order of enzyme. Okay, this is how the enzyme are naming. Okay, now comes to classification of enzyme. Okay, in the earlier time, I'm talking about in the previous, that is earlier time, the enzyme are classified on the basis of reaction they catalyze. Okay, please do remember, in the earlier time, okay, on the basis of reaction they catalyze. For example, when you're talking about proteases, that is proteases, then this enzyme catalyze the protein. Okay, now when talking about nucleases, then it also catalyze the nucleic acid. Okay, now some example, that is hydrolases, then it catalyze the reaction that is digestive enzyme. Okay, now this is how the enzyme naming are enzymes are classified. Okay, in the earlier time, but nowadays, please do remember. But nowadays, there are total 6 class divided into 6 class. Okay, please do remember. Enzyme is divided into 6 class. Okay, out of which contain that is 4 to 13 subclass. Please do remember, enzyme is divided into 4 class, and out of which it contains 4 to 13 subclasses. Okay, what are the 6 classes? Please do remember, simply you have to learn that is othlil, OTHLIL. Okay, simply you have to learn othlil in a very sequential manner. Okay, why? Because it is defined as, okay, first order, that is O stands for oxidoreductase, which is a class 1. Okay, this is a class 1. And second class, that is transferase. Now, this represents second class, that is hydrolysis. This is a 3 class. Then the ligases, it will be 4th class. Then isomerases, that is a 5th class, and the last one, that is ligases, that is a 6th class. Okay, now let's see the detailed account of auxiliary, that is class. Okay, 6 classes. Let's proceed further. Let's see. First class, that is oxidoreductases. Okay, it is also called dehydrogenases. Okay, that is class 1. Okay, class 2 is, that is transferases. Okay, this is class 2. And the third class is, that is hydrolysases. Okay, and the fourth class is, that is lysases. And the fifth class is, that is isomerases. And the last class, that is sixth class is, that is ligases. Please do remember. Now this is the sixth class. Okay, now let's see the oxidoreductase. When you're talking about oxidoreductase, then the enzyme catalyze, please do remember, enzyme catalyze the oxidation reduction. Okay, enzyme catalyze the reaction. What kind of reaction? That is oxidation or reduction. You have to remember, oxidation and reduction. Oxidation and reduction of substrate. Please remember. Okay, now this is called oxidoreductase. Okay, for example, let's see. It become reduction and it become oxidation. Okay, this is how the reaction after. Okay, but when you're talking about transferases, please do remember, then enzyme catalyze the substrate. Okay, enzyme catalyze the groups. Okay, please do remember, enzyme catalyze the group that is other than hydrogen. Please do remember, other than hydrogen. Okay, that is a common sense. Okay, when talking about the gaining of hydrogen and removal hydrogen, that is the oxidation or reduction. That is coming to the category of oxidoreductases. Okay, but when you're talking about transferases, then enzyme catalyzing the group, okay, other than the hydrogen, that is called transferase. Okay, for example, let's see here, that is now it become, okay, okay, let's see, that is group is transfer, okay, other than hydrogen, this is called transferase, that is class 2. But when you're talking about hydrolases, please do remember, hydrolases, the enzyme catalyze the reaction, okay, that is other than, please do remember, simply it catalyze the reaction, okay, that is peptide bond, ester bond, or ether bond, glycosidic bond, etc. Okay, now what we are talking about ligases, that is class 4, then it simply, okay, that enzyme catalyze the breaking of, please do remember, breaking of breaking the bond. Okay, what kind of bond? Please do remember, for example, let's see, in this case double bond will be restored, double bond will be restored. Okay, let's see, for example, that is, let's see here, carbon make the bond double, that is X and C, and this carbon also make the double bond. Okay, but we are talking about X and Y, then it make a single bond. Okay, so the double bond will be remain as double bond will be restored. Okay, and becomes the, that is X and Y. Okay, now this type of reaction is called, come into the category of, that is class ligase. Okay, but when you're talking about isomerases, then it simply catalyze the reaction of optical isomer, that is geometrical isomer, structural isomer, position isomer, that is come into category of isomerases. Okay, but when you're talking about ligases, then it simply join the two bond. Okay, it simply, please do remember, join the two bond. Okay, for example, in case of DNA fragment, okay, that is Okazaki fragment, the two bonds are joined. Okay, that is called, come into the category of ligases. Okay, now this is how the classification of enzyme is completed. Now let's proceed further, that is cofactor. Okay, now let's see. Cofactor. Okay, first of all you have to know enzyme is divided into two categories. Okay, enzyme is divided into two categories. Okay, one is called, that is simple enzyme. A second one is called complex enzyme. Okay, now we're talking about a simple enzyme, that it is entirely made up of, please do remember, entirely made up of protein. Okay, as we all know that, as I was stated earlier, कि enzyme contain different kind of configuration of protein. Okay, that is protein contain different kind of configuration. That is primary structure, secondary, tertiary, and quaternary. Okay, so entirely when talking about enzyme, the enzyme is entirely made of protein. That is coming to the category of simple enzyme. Okay, but when talking about a complex enzyme, then it is made up of two subunit. Okay, it contains protein as well as non-protein part. Okay, protein part is called apoenzyme. Please do remember, it is called apoenzyme, and the non-protein part is called, that is, cofactor. This is called cofactor. Okay, now let's see. The complex enzyme is called, for example, let's see, complex enzyme. Complex enzyme is called holoenzyme. Please remember. Complex enzyme is called holoenzyme. Okay, now the protein is made up of protein plus non-protein. Okay, protein part is called apoenzyme and non-protein part is called cofactor. Okay, now let's see the protein part of enzyme. Okay, now this is how the complex enzyme is divided. Okay, so let's see the cofactor. Okay, now what are the cofactor? Okay, so the cofactor is divided into three categories. Okay, three different type of cofactor are found. Okay, it may be prosthetic group. Please to remember, that is prosthetic group. Okay, it may be coenzyme. Okay, and the last one is, that is, metal ion. Okay, now when you're talking about the prosthetic group, okay, then it is made up of organic compound. Please do remember, it is made up of organic compound and tightly bound to the apoenzyme. Okay, please do remember, and tightly bonded to the, okay, that is apoenzyme. Okay, for example, let's see here. For example, that is talking about that is catalases, peroxidases. Okay, peroxidase catalase, that is H2O2. When act peroxidase in it, okay, then it form water molecules plus nascent oxygen. Okay, now we're talking about a heme, that is, okay, let's see. Heme is the part of prosthetic group. Okay, part of prosthetic group. Okay, which provide the active of apoenzyme. Okay, now this is how the prosthetic group. For example, let's see, when you're talking about it, that is organic compound and tightly bonded. Okay, to the prosthetic group, is tightly bonded. That is apoenzyme. Okay, but when you're talking about a coenzyme, please do remember that it is also made up of organic compound organic compound, but it is transiently bonded. Please do remember, bonded to apoenzyme. Transiently means it is weakly bonded. Please do remember, weakly bonded. Okay, for example, let's see that this is the structure of apoenzyme. Okay, then it will be weakly bonded. This is how. Okay, now that's why it serves, please do remember, it serves the number of reaction which catalyze. Okay, that's why because it loosely bonded. Okay, for example, let's see, that is vitamin NAD, that stand for nicotinamide adenine dinucleotide. Okay, and NADP, which contain vitamin niacin. Okay, now but we're talking about metal ions. Okay, please do remember, talking about metal ions, then it form the coordinate bond. Okay, it form the coordinate bond to the apoenzyme as well as the substrate. For example, let's see here, this is the apoenzyme and this is a cofactor, that is metal ion. It will bond like that, that is coordinate bond. I am showing here coordinate bond. Okay, and the same time it make bond the substrate also. Okay, this is how the metal ion work. Okay, for example, that is carboxypeptidase. Okay, now this is how this chapter is